Good day, class. Thank you so much for connecting and subscribing to Metric Hub. I am coming to you with a very quick and basic tips towards your paper two that is coming on Monday. I'm going to give you a quick rundown of what you should prepare and make sure that this is what you should get ready for and you will never go wrong. Just keep connected without wasting any time. Please note that the first thing that you are going to, that you cannot go to exam not knowing is the uh, life at a molecular level. So in this case, I'm just going to be touching base and reflecting to you what you should prepare in each of the areas. So I mean, like here you should understand uh, DNA. You should know DNA structure. Here, in terms of this, obviously, we are saying on the DNA structure, you should know the structure of DNA, that it is a double helix. DNA is a double helix. You should understand that you should know the pairing of bases, the complementary pairing of bases, complementary pairing of nitrogen bases. This I remember, you remember very well that we talked about A and C. I mean, A pairs with C with T, and also we have got G. It pairs with what? With the C. That is the complementary pairing. And then still on that, please note that again that there are questions which comes on multiple choice where you are asked to calculate the total number of any of the nitrogen bases that is given. In this case, please note that you will be given the number of any of the nitrogen bases. Let's say, for example, you are told that A has got this nitrogen, has got this number of nitrogen bases, and G has got this. So what I want you to take note of is that the number of A is always equals to what? To T. That is one thing that you should know. And same applies. And G is always is always equals to is always equals to C also. Is always equals to C. So you know that C and G, they pair up together. The numbers of these nitrogen bases should always be equal to each other, A and T, as well as G and what and C. So if you are given any of them, you should know that in the total number of bases, the total number of nitrogen bases is equals to A times 2 plus G times 2, which is going to give you the total number of nitrogen bases. Total number of nitrogen bases that you get there. The reason is because why we are saying times 2 is because it's A and T and it's G and what and C. That is what we are meant to say there. So that question, if ever it comes, you need not to get any mistakes there. And then still on that, you should know the functions of DNA. Functions of what? Of DNA. I'm sure that this you know that DNA, what is the function of DNA, and then it controls the structure and functioning of organism. It is the one that also allows the formation of mRNA during transcription. So that is fine. You should know the location and types of DNA that you must know. You must know that the location and types of DNA, I'm sure that you know, which we can say that we have got what? We have got nucleus DNA, which is the type and location again. We have got mitochondria DNA. We have got mitochondria DNA, and then we have got the third one, which is what? Chloroplast DNA. This is the types and location. Those are the three types of DNA that we have. Those are the three types of DNA that we have. And then still on that, I'm saying still on that, you need to understand, you should understand the difference. Sorry, before that one, I'm sure that you should understand that you should understand the building block of DNA, the building block of DNA, which you remember that it is what? It is the nucleotide. It is the nucleotide, nucleotide is the building block of DNA. I'm sure that you know, wherever you are given that, you know very well that the DNA structure is something that we can always know. That is something that you should know. The nitrogen base, the phosphate, and the sugar. Remember that the sugar is what? Is deoxyribose sugar. It has got deoxyribose sugar. That one you should know. Sugar is deoxyribose. That one you should know. And then you should you are aware that that is the DNA, what you call building block, the nucleotide. This diagram, it might come also, and you'll be asked to label it. This is a nucleotide, and you need to know to say what is A, what is B, and what is C. That you must understand. You cannot go to exam without getting to know that. I'm sure that you know. Let's go to the next question. The differences. The differences are between DNA and RNA. Remember, this question, it normally comes as to say tabulate. Normally the question says, it says tabulate the difference. Tabulate the difference, tabulate the differences. So what you need to do is you need to draw a table which you can give to say DNA, RNA, and then you give the differences of these two to say DNA. DNA is a double helix. It's a double helix. RNA is a single strand. RNA is single strand. I'm saying two. You can say DNA contains deoxyribose sugar. It contains deoxyribose sugar, and then RNA, it has got a ribose sugar, and then ribose sugar. We can also, number three, say that contains thymine, nitrogen base, contains thymine, and then RNA, it contains uracil instead. That is some of the differences that you need to take note of, the major differences. So there is no pairing of the bases in RNA, and then obviously in this case you go to types of RNA. Types of RNA, that one you must know. You must know the types of RNA. We have got one, the mRNA, which we said that it's formed during transcription. It is formed during transcription, this one. And then you must understand that the mRNA, it contains what? It has got the nitrogen bases, which are called codons. 3 nitrogen bases that are found there. And then if you look on another type, if what? tRNA, which we are saying that it has to bring the complementary bases, which are called the anticodons. It contains the anticodons. And then in terms of that, obviously, you should also give their functions of these RNA things that we are talking, molecules that we are talking about. Having said that, let's go, you talk about DNA replication. You can't go to exam not knowing about DNA replication class. DNA replication, a process by which DNA makes a copy of itself. where we are saying a small piece of DNA unwinds and the two strands are used as templates for the formation of what? Formation of another strands which are formed through the what? The nucleoplasm, which is containing the what? The nucleotides that we're talking about. So DNA replication, that one you must know the definition, need to know the definition in the process of how does it occur. You should be able to describe it. I go again, you have to talk about the process of transcription. You can't go to exam not understanding transcription class. Transcription is very important when a process where mRNA formed. mRNA is formed from DNA. You must not forget that I'm just going to give you a rough diagram again of this, which you should take note of there to say that when we are going to answer this question, you must know very well that the process of transcription, it has got to do with where you are going to see a small piece of DNA which is blending in between the strands of RNA. This is called the mRNA that we are talking about, which is formed by what? By the free-floating nucleotide bases and stuff that are in there. And then this diagram, you always know that we have got, it's a small piece of DNA that unwinds, and as it unwinds, we are saying this area which we are seeing here is the where the DNA it is unwinded, and then you must know that how do you know that this is transcription? You see it by having a single strand of what? This is the single strand of mRNA that forms, and then it lies outside, it lies alongside on its own there. So sometimes this question, it comes where you would also see some diagram like this, which is going to show you the nuclear plasm to say label A there, and then we are saying is what, which is the nuclear pore. The nuclear pore, you also see that we have got the cell membrane, we have got what and what. So transcription, you know that like you know it like that. So what we are saying is that whenever you see a single strand of RNA formed, it is what you call transcription. It is a process through which mRNA is formed from DNA. In this regard, a small piece of mRNA, it blends itself, it is formed, assimilating the information from DNA such that it will contain the direct information or the duplicate of the information that is contained in DNA. So what you need to understand is that there is still complementary pairing of bases that takes place in there to say that I can give, for example, that let me say, we can say we have got bases A, G, C there. So we need to see what is the complementary basis that can be found on what? On this mRNA. So you should know that opposite to A, we have got U, and then we have got C, and then we have got G as the what? As the complementary basis. So on DNA, we have got these bases. I'm saying we have got A, G, C, and then on mRNA, that is coming with its complementary basis, I'm saying you should know that it comes with what? With U, G, UCG, because remember, U is substituting what? U is substituting T. Instead of T, we have got U there. So this is the complementary pairing that you should not forget in that case. This is the complementary pairing which every learner who is going to write on Monday must not miss. You mustn't miss this. You mustn't miss that, this pairing of the bases. Having said that, I'm quickly just rushing to make sure that you get everything that I'm expecting of you. You need also to talk about protein synthesis. protein synthesis. Protein synthesis is a process by which proteins are formed. We are saying a process through which proteins are formed, which we are saying it is also called what? Which is also called translation. It is also called translation, which I'm saying is a process by which proteins are manufactured, step by step, step by step, through ribosome. When it reads the order of mRNA, ribosome reading the specific order of mRNA codons. So the order of the mRNA is what defines how the proteins are to be manufactured. So please, class, don't make a mistake there. You should understand that the process of what is this? You see it by you are going to see the codons and anticodons there. The moment you see those, you should know that that question, it is asking you to talk about, to know that this thing that you are to talk about is what is the process that you are to talk about is called what? It's called, I'm saying this is mRNA, and then we have got that is called the ribosome, and then I'm saying it is the manufacturing site of the of the proteins. So what we are saying is this, when we are saying these are called codons, which are called the 3 nitrogen bases found on mRNA, and then we are going to have opposite to it, we have which we are going to call this, we are saying on this other side, we have got what is called the anticodons. The anticodons are found on what? On tRNA. And then don't forget to say we have got what is called the amino acids, which are brought by the what? By the tRNA. And then what you need to understand is that these codons, I'm saying they are from the mRNA, and the anticodons are from the what? From the tRNA. So please take note of that. So what happens is that this mRNA, it is going to pass through the what? The ribosome. The ribosome is going to read the order of this one, CCA. So CCA, it is we are saying as it moves there, as it passes through the ribosome will read this CCA, and then the anticodons are activated the tRNA is supposed to bring the corresponding anticodons, which is going to bring what? U, which is going to come. So therefore, it comes with the corresponding amino acids there, and then specific proteins are manufactured. In that case, specific proteins are manufactured. So what I'm saying is, when you see a question like codons and anticodons, the process taking place, it is what? It is the protein synthesis, that you must not forget that one. Having said that, don't forget about mutation, gene mutation, still on this topic. Don't forget about gene mutation. Gene mutation, we are saying mutation, when it takes place, it results in the formation of a different proteins than what has to be formed. So mutation can take place, which can affect our what? Which can affect the order of our nitrogen bases there. When mutation takes place, a different amino acid will be formed, which sometimes if that mutation took place and then the same amino acid it formed, it means that the mutation has no effect and no harm can happen because we still have the same amino acid being formed. So please don't make mistakes there and make sure that you keep on understanding and not getting it wrong, whatever that you are going to come across during this examination. So that is about the what? About what called DNA and stuff. I want to bring to you, let me talk about another topic that I want you to take note of. Still under paper two, still under paper two, what we need to take note of is this we need to talk about genetics and inheritance. Genetics and inheritance. In terms of this question, this topic, most important here is that you need to understand that uh, inheritance, we are talking about the passing on of genetic characteristics from one generation to another. And then it is caused because of what? Because of genes. These genes are carried by the chromosomes. So in this case, one key area that I want us to take note of in this topic, the NB or key topic, key specific areas that you guys must not forget is that you can't go to exam not understanding a genetic flow diagram. A genetic flow diagram, you see, when we're talking about the inheritance of characteristics, we either use a genetic flow diagram or we use a Punnett square. That you must know. So let me start with the genetic flow diagram. This is a diagram that illustrates how genetic characteristics are inherited. So through a genetic flow diagram, we say P1, and then we say phenotype. Phenotype, we are talking about saying, for example, phenotype, we are talking about the outward appearance of an organism, the physical outward appearance that we can see. So we can say a tall pear plant is crossed with a short pear plant. So in that case, we are saying that is what you call the phenotype, and then you should know the genotype. Genotype, we are talking about the genetic makeup which is found in the blood. So let me say these are homozygous plants. So the homozygous plants, we are saying this is a homozygous plant, and then obviously the also homozygous plant which is short. So genotype, phenotype, and then you should know that there is what is called meiosis. Meiosis, it is the process that results in the formation of gametes. Gametes, meiosis, you know this, the principle of meiosis, it halves the number of chromosomes. So in this case, you should know that when meiosis takes place here, we have a single T. And then when meiosis takes place there, we also have a single recessive T. And then the next step is what is fertilization. Fertilization is the fusion of the sex cells, which we are going to take this T here, it comes there, that small T, it comes there, and then we have got that T. We have got that heterozygous T. So this is called a genotype again, which means to say that the genotype is like that, and then we say the phenotype. The phenotype again, we have got what? We have got 100% tall plants. So this is what we call our F1. Our F1 is the first filial generation that we are talking about. So this diagram class, there is no way which you should go to exam without understanding it clearly and as much as you should know it by heart. This is a genetic flow diagram. This is a genetic flow diagram. So we are saying in terms of this, we have got this called what you call the monohybrid cross. This is a monohybrid cross, which you know that is one characteristic involved. So monohybrid cross F1 of monohybrid is always 100% of the dominant allele. F1 of the hybrid of monohybrid cross is 100% of of the dominant allele in question. So as you can see here, we are saying this we are talking about F1, which are pure breeds. In this case, we are talking about pure breeds class. Pure breeds. We are talking about pure breeds, which you need not to forget that pure breeds are also called what? Homozygous. They are also called homozygous, which means to say they are of the same. We have got the homozygous, as to say dominant T and also recessive T. I'm sure you still remember that we said when we have this, we said this is called a dominant allele, and then that is called recessive allele. So the dominant allele, it takes 100% of that. Having said that, I can as well tell you that let's calculate, we need to calculate the F2 generation, the F2 of monohybrid. F2 of monohybrid is what? It's always having is 3:1, which I'm saying it is what? Or 75% as to 25%, which we are saying 75% it is from the dominant allele, and then 25% is to what? Is to the recessive allele. That you must not forget as well. You must not forget that. That must be known by every learner who is going to write metric. Remember that we can as well prove, but I'm sure that, let me not go there because of time. I just want you to remember that this is called a what? A monohybrid cross. F1 is 100%, and then F2 is what? 75% as to 25%. That you need to know about this. I'm again, having said this, let me just again explain to some of you who does not understand, to say, where are we getting F2 generation? How do we get F2? F2 generation, I say that F2, when we are doing F2 generation, when we are doing F2 of a, when we are doing F2 generation of what? Of monohybrid, you should understand that we start with P2. We say P2, and then we say P2 means parents 1. We are taking the offsprings of what? Of F1, of P1. So we are saying P2, we are going to say phenotype, and then we say genotype, we say meiosis, we say fertilization, genotype, phenotype again. This diagram, it doesn't change. So I was saying phenotype, we've been talking about, it is a tall plant that is crossed with a tall plant. So the genotype, we say that it is heterozygous T is crossed with a heterozygous T, which we are saying meiosis, we say that the gametes must not be forgotten. So we would have T, and those T, because we said that meiosis, it halves the number of chromosomes. So we have this, these chromosomes as to what? As T, and then T there. You understand? So fertilization, it is when we talk about this T, it comes here, and then it goes with that T here. Then we have got this T. And then we say this T again, it comes there, it goes with this other small T there. we have got this T, we have this small t, it comes here and then it goes with the other big T. So we have got this T and then this T, it goes with the small t there. Then we have got those three small t's. So there we are saying these are different outcomes that we are having. Hence we have got this, we have got to say the phenotype. The phenotype, you count all the toe. We have got toe, 1, 2, 3. This is 3 toe is to 1 short. That is where we got the 3 to 1 thing that we talked about there. 3 toe to 1 short. Hence, we have to say that 3 toe to 1 short. We said as a percentage is chance factor 3 over 4 multiplied by 100 over 1 is to 1, this one 1 over 4 multiplied by 100 over 1. So we get 75% is to 25% of what? Of the F1 generation. This, I mean, of the F2, sorry. This is our F2 of the, of what? Of monohybrid cross. That is F2 of monohybrid cross. So as you do that, you won't make mistakes and then it will be done. This you won't make mistake and then you will know very well that you have got this correct. And having said that, I was saying there is what is called a Punnett square, which I said that the Punnett square, it also, when after you reach to gametes, I said that your gametes, you have got your gametes, which I said that the gametes are what? We have got our gametes as T, we have got our gametes as T, and this T, which is crossed with T, and that T. So with a Punnett square, when we are talking about a Punnett square here, we take our gametes from this point. We take our gametes from that point where we are going to draw a diagram which gonna be used to calculate this. We draw our diagram there, which we can use to define what we want to calculate here. So I'm gonna show you now to say we have got our T and then our small t there. We have got our T and our small t there. So what we are saying is these are what? These are gametes, which we can say male and female. These are our gametes that are shown there. So having this, what we're supposed to do is we are taking this T here, it comes here, and then that T there, it comes there. We take also this T here, and each of them goes with what? With what is coming according to the table or box that we are having. We take this T here and that T here. We have got this T and this T there. We have got heterozygous T, and then we have got this T, recessive T together. We give those three T's. So same applies. It comes back to say we have got 3 toe. It comes back again where we are going to have 3 toe as to 1 short from our Punnett square that we are working on there. And then it brings us back to the same calculation that I have shown here again on the board, which I have shown you to that. This is a Punnett square. So Punnett square, you use it after you have reached to the point of what? Of gametes. That is one thing that you should know. That is one thing that you should know there. And then let's go on still on these calculations. Still on these calculations, I want to show you another question again, which you should also take note of, which we call dihybrid crossing, which we call dihybrid crossing. Dihybrid crosses. We are saying this we refer to two characteristics. are considered at a go. Two characteristics are considered or involved in the cross. So I've been talking about this data. When you look at when you are looking at this, you should know that dihybrid crossing, it is always something that you should know to say that it starts from that question where we've been talking about purebreds versus what I'm saying, a purebred that is dominant versus a purebred that is recessive. So we are saying we have got our plants, which we can say that it is green and ringwood is crossed with what? With yellow and round. Green and round. Let me say green and round. It is pear plants that we can say it is green and round seeds are crossed with what? With yellow and ringwood. Yellow and ringwood seeds. So in that case, what you should take note of is that we remember to say you always have a key that will define to say what are you referring to say as green. So we can say that green is represented by what? By capital letter Y. So green is represented by capital letter by dominant Y, which we say capital letter Y, and then round is represented by R. We are saying this is a purebred, and then it's crossed with what? With yellow and ringwood. So yellow is represented by recessive Y, and then ringwood is represented by recessive R. So in this case, remember to say, you can be asked to say what gametes, number of gametes that can be produced here. So we are going to say in this case, meiosis, we are saying when meiosis occurs in this regard, what will happen? We have got one Y there and then one R because we have got two Y. So we have got that, and then it's going to be crossed with what? With one Y and small R also there. These are single gametes, which we can say they can be crossed together. So fertilization will occur where we are going to have Y and then R like this. Having this, it would mean that we have got what? Yellow and round. We are going to have yellow and round seeds as the outcome that we can get in this case, as the outcome that we can get in that regard. So what I want you to understand is that dihybrid crossing at F1 generation of it is 100% also of the dominant alleles that you see that we have said green is dominant. We said that green is what? Green is dominant. And then also round is what? Round is also dominant. Round is also dominant. So the dominant alleles are the outcomes that we can get at the end of the day. So when you are to calculate here in this case, you need also to know, like to say, we are having now yellow and round seeds as our outcome, which we must use to calculate our what? Our F2 generation. So our F2 generation must be based on how many, you need to know the gametes that must be formed here. Let's say, in this case, we must find out the gametes that must come out of this. How many gametes should we have out of this? So in terms of the gametes, eh, please note that I have given it also on the channel that you always have to understand that uh, these these gametes or these letters are known that we are giving here. They can never be of the same. I mean, we cannot have Y alone in a single gamete. We cannot have YY in a single gamete, but we must have what? We must have a mixture of we must have a combination of both of these. So in regard to that, let's say we are saying we have got R. So our gametes that we can have one, the first one, it can have this Y, where we have going to have Y and R dominant, or we can have also this Y, we can have a dominant Y, and also we are saying dominant Y and small r, that is the second one, or we can have small y and r, we can have r, y, or we can have uh, we can have also the recessive alleles on their own again. We can have also the recessive alleles that are on their own, which can mean to say that we can have also, we are saying or we can have YR. So we have got four gametes that can be formed here. We have got four gametes that can be formed. Four gametes that can be formed in this case. These four gametes, you can use them to do your Punnett square, which we are now going to calculate your F2 generation. You can also use it to calculate your F2 generation, which I said that you can also use it to calculate the F2 generation, which you should be able to do. I can just want to quickly show you that it is possible to do that. How do you do that? We are saying you have got your Punnett square, which you can draw as usual. One, two, three, and four. We have got one, two, three, and four there. So what we are saying is that our gametes are these ones. So I can take YR, I'm taking this one. I can take the next one, YR, this one. I can take RY, and then the same again, small y and R. It's like that. Here the same thing, I can have R, YR, I can have YR, I can have also RY, or I can have Y like that. So I can give this to say that I can say male, and then I can say female, which is also female, which is this direction. So when we are crossing them class, you just take whatever in this box versus whatever in the next box. So I can say this is Y, and then this is Y, this is R, and that is R. So I'm taking everything which is in each of the boxes. Y here, and then there is another Y there, there is R there, and then there is R there. I'm taking this capital letter Y, dominant Y, and small y there. I have got this dominant R and that dominant R there. The same applies. I can take this dominant Y and the small y, which is there. I have got the dominant R and small r until the whole box is completed. Until the whole box or all these Punnett boxes are given. So you can do and fill up that, and then you can be able to calculate what? You can be able to calculate the chance factors of each of them. I can say, let me do this one just to make sure, just to demonstrate to some of you. I've got this y, capital dominant y, and the small y there. I've got this dominant r and the other dominant r there. I've got this dominant y and small y there. I have the dominant R and small r. I have got this small y and small y. So it's small y. It's kept is dominant R and dominant R. I have got this dominant, I mean this small y and small y there. There is this dominant R and small r. So we do like that until the full unit is completed. So just to present to you again some of the key areas that you should know. You should just be able to interpret the meaning of these. So in terms of this, I can say this would mean yellow round box. Now this box, this one, it will mean that we take each of the combination of the alleles that are there and interpret them. So we can say that means yellow round. It's yellow and round seeds. This one again is yellow and round. This one is yellow and round. I can come to this one. I mean, it's uh it's yellow and round. I can come to this one, which is green and round. I'm talking about this one. If I can take this one, I'm just trying to uh interpret for you to know, so therefore you can know. So I'm saying that box that I've shaded there, it will mean green and what? And round, as you can see from those. So you have to read all of them which are green and round. You read all of them which are yellow and round, and then you make sure that that is done. You make sure that is done there. So let's just go on to the next area that I want you to take note of. I was saying uh, we have got sex determination. Sex determination, naturally we are saying is the chance factor of getting a baby boy or a baby girl. That one, it is always a 50-50%. It's a 50-50 percent, because remember we have got what is called sex chromosomes. Sex chromosomes, which we said that we have got, for male, we have got XY, and then for female, it is what? It is XX. So we are saying the chance of having a baby boy or a girl, it is going to be saying X, and then Y is crossed with what? With X, and then we are going to have this X and that X there, we have what? XY, XX, and then this Y and that X, we have got X, XY. So it is one boy, I mean one girl, as to one boy, which is why I said that it's what? It's a 50-50%. One boy as to one girl. XX, it means what? XX, it means a girl, and then XY, it means boy. So it is one girl as to one boy. And then let me quickly just bring to you again to say that they might be given that the family is already having children. Some of those questions, therefore, I remember I once showed it on the channel. If the family has got children and they are expecting the probability of them having a baby boy or a girl, I said that is a basic mathematical question. You don't go through it like this way. Let me go to sex-linked diseases. Sex-linked diseases. These ones, I said that you should know that we have got what is called, I don't want to go into much of this. You need to know these are disorders that are not so dangerous. They don't cause death, but might affect the functioning and performance of an organism. So let me talk about male and then versus female. In terms of this, we have got those diseases, color blind, hemophilia, and so forth and so forth, so forth. So in that case, I'm saying we have the genotype and then we have the phenotype. To say in male, we remember to say the most of the times these diseases are caused by a recessive allele, but sometimes the question will give you what it needs you to understand. So for instance, I'm saying for male, we have got only two options. Man is what? We're saying it's X, man is XY. So if a man is having a dominant C, I'm talking about color blind, this will mean to say this man is what? Is normal. However, if it is X and the recessive C, we are saying this man is what? Is color blind. The man is color blind. The man is color blind in that case. And then same applies in female again. So we have got two options, a normal and a colorblind man. That is one thing that you should know. And then in female, we have got we are saying the genotype. The genotype is like to say we have got XX, and then if it is XXC, it means it's a normal female. And then we can have X C. This will mean a carrier female. And then we have got XX, and then small c all together. It means it's what? A colorblind female. So what you need to know is that these things, whenever you are given questions, you should know that there is what you call a normal, a carrier, and what? And an affected, and an affected female. This must know that this is a small c. This is a small c here. That is what we are saying there. And then one other question on this topic is that you should note that we have got what is called uh, the diagram, which we use to say that the pedigree diagram, or it can be called a pedigree diagram or cladogram or something, which you are supposed to understand that whenever you are given children whereby the mother and the father, they look affected, they look normal from the shaded from this, we are saying it is a mother and father, and then they give birth to children who are they are giving birth to children who are, one of them is affected. So if it is like that, to say that one of the children is affected, it means that will mean to say that the what? The mother is a carrier. The mother is what? Is a carrier. So when you're going to do your calculations, don't make mistakes of that. Make sure that if ever these cycles or shapes are not, these formats are not shaded, but there is an affected child in these people, it means that there is what? There is there is an affected boy child. It means the mother is a carrier because the child has inherited a recessive allele from the mother, which is from this, which I gave you to say it will be like that. Quickly again, you can also talk about blood groups. Blood groups. We have got ABO blood typing, ABO blood typing. That you should know that it gives us 4 blood groups. It gives us 4 blood groups. These are to say blood A, blood what? AB, blood blood B, and blood O. And understand again that these are 4 blood groups. They are controlled, all of them are controlled by 3 alleles. These 3 alleles, they are allele A, allele B, and allele O, and understand that allele A and allele B are dominant alleles, while O is recessive. Hence, we can only have a child having blood type O if uh, I and I have crossed each other or are combined together. That's one thing that you should know. And then in terms of blood groups, I just want to let me give an example. Example, example one, we can say that in terms of this example one, what I can say is, let's say blood group, blood group A. We want to give the genotype. This is the phenotype. So the genotype of this blood group A is equals to, we can have blood A as to say it can be AA like that, or we can have blood A being a combination of what dominant dominant I and A and O. So the same applies also, or B, we can have also B like this, or it can be also B and I, whereby we are saying this B is dominant and this A also is dominant. Hence we can have that blood type B or A. So understand in this case that uh allele A and B are codominant. Allele Alleles A and B are codominant. They are both expressed in what? In blood type. And then having said that, blood type is not conclusive in terms of paternity testing. Why is that so? Because it can only exclude a father from being a father of a child, but cannot confirm that somebody is totally a biological father of a child, because people can share blood type. One blood type can belong to more than a million fathers. So that is one other thing that you need to take note of in that case. Uh, so that is one other thing. Still on scope, still on scope. Let's talk about another area that you should take note of in terms of paper two. You should note about speciation. Speciation. I said that we have got geographic speciation, uh, and also we have got what is called reproductive speciation. So having said that, on geographic geographic speciation, you need to know that there is a physical barrier involved in this case. A physical barrier is involved. Example, e.g., we are saying it can be a mountain range. It can be a mountain range. It can be a golf course. It can be a desert. It can be a water body, which can be a sea or a river. uh, any of such a nature which we can say it's a physical barrier. So quite often you should know the process of geographic speciation, which we are saying organisms are separated by a what? By a mountain or a water body, which can cause them not to interbreed, and therefore they cannot be able to, there is no gene flow between the two populations, which results in hybrid sterility if either those population groups are to meet again. So we are going to have what you call the two population will be genotypically and phenotypically different. So that you need to note all of those in terms of speciation. And then the reproductive isolation, reproductive isolation, reproductive speciation, it talks about every means that can separate two populations from breeding together. It can be breeding at different times of the year, feeding at different times. It can be gametic isolation, courtship behavior. All those they fall under what? Under reproductive speciation. So the process of geographic speciation, it must be well known. It must be well taken note of. So if ever you see a population species, we are seeing a population species which is moving, and then certain point there, we are seeing it is going to be A, and then we are going to have B, and then that forms there, we are saying population A, and then population B, and then this is a mountain range. Automatically know that this is geographic isolation. This is geographic isolation. So the mountain range, it stretches to a point whereby even if this will come, they will move, but they'll take million years to come to this. But we can have what you call hybrid, hybrid sterility, which will result at the end of the day. So that is what is happening there. And then having that, I want you to look in what is this evolution, the theories of evolution. We have got the Lamarck theory, Lamarck. We have got also Darwin's theory. And remember that the Lamarck theory was rejected. Lamarck's theory was rejected, and Darwin's theory was what was accepted. We need to know the principles of each theory. I was talking about Darwin's theory, for example. Darwin's theory, remember that it was based on natural selection. It talks about variation. It talks about great population. I mean, organisms giving birth to a variety of organisms. I mean, a variety of offsprings. It's all those things. So the principles of each theory must be known. And then I'm saying uh, reasons for the rejection, reasons why Lamarck's theory was rejected. Reasons why Lamarck's theory was rejected, that you must take note of that, which we said that it was too deterministic. Organisms could not were evolving because they want to evolve. And then the theory also, it was talking about inheritance of acquired characteristics, which is not possible. So we are saying reasons for Lamarck's theory to be rejected, that must be taken note of. What is next? What is next? What is next? Still on the theories of evolution, I want you again not to forget that you should know, you should take note of, you should take note of other theories of evolution, other theories of evolution, which we're talking about punctuated equilibrium, and also gradualism. In this case, you should also be able to explain the two theories, and also NB, the graphical explanation of the theories, and then graphical explanation of the theories must be taken note of. In that case, you need to know that you need to note also very well that for example, we need to understand the issue of the why an insect can be a best example in explaining uh, these theories, in explaining these theories. That's, for example, e.g., mosquito. I'm sure that you can see from the previous question paper of preparatory this year, I explained that there is a good explanation for that question. Please take note of that. And then still on this question, on these sources of evidence of evolution, sources of evidence for evolution, they must be also taken note of. We talked about this biogeography, biogeography. We talked about genetic similarities. We talk about modification of body plan. We talk about cultural evidence. We talk about what else? Cultural evidence. We talk about embryonic similarities. They are good examples in terms of explanation of sources of evidence. Cultural, we are talking about the way life goes on, and not to forget the biggest one, fossil evidence. Fossil evidence, it must be explained. How are these working in terms of explanation of evolution? And then, maybe last but not least, you need to understand uh, the out of Africa hypothesis. Out of Africa, out of Africa hypothesis, out of Africa hypothesis. This has to be understood to say what explanation is there that supports out of Africa evidence of out of Africa hypothesis, evidence of out of Africa hypothesis, and then what happens if out of Africa is disputed, if we can say that the evidence explains explaining about human evolution is found in Asia than Africa, therefore out of Africa hypothesis is disqualified, and then this must be taken note of. I'm sure you need to also not to forget the phylogenetic or evolutionary diagrams. Remember, these diagrams, they explain the way through which evolution took place. So we are saying organisms that appear to be close together, they are what? They are more related than those that are far apart. And then we are able to understand also the million years ago, which organisms existed, where we need to explain also the extent versus extinct, extinct versus extinct species or extinct organisms, extinct organisms. So what I'm saying is, having done this, family, you won't go to exam and don't get the best out of life science. I saw what happened in paper one. The examiner tend to be too much focused on evolution. I mean, too much focused on reproduction and homeostasis and stuff. But let's expect that in terms of paper two is the one that we are going to boost. Thank you so much for your time. Don't forget also under evolution, you are going to understand and read about the hominids of Africa, the hominidae from the Australopithecus africanus to Homo sapiens sapiens. All those you need to know them. The differences in terms of their foramen magnum, in terms of the pelvis, in terms of the bipedalism, in terms of the brain capacity, the cranium size, how does it affect evolution, the detention of teeth and stuff, how does detention of teeth was affected by the discovery of fire, all those class, I'm sure that they should not be just be ignored. Go to exam and get the best. I wish you all the best. Thank you so much for your time. And this is the scope, and you will never go wrong with this. Just refer and check on if you are getting lost, what you need to get to look at. Metric Hub is there to support you. We meet on Monday to celebrate and get the distinction in life science. Metric Hub loves you with the love of Christ. All the best until we meet again. More is still coming. And just refer to your brothers and sisters. Just to know that Metric Hub is there, is there for them. Thank you so much. Let's keep on bringing them to subscribe, and no one will have the same performance and will remain walking this journey alone. Thank you once again.